Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we interview authors from our feature articles, the papers of the month, or other articles published in that month's print issue of the Diabetes Journal. We also have a Sweet Talk segment where we get a little creative in our scientists and topic interviewees, and we have a good one for you today, as usual. My name is Darlene Sandoval, and I'm joined by my podcast co-host, Dr. Kirk Haberger and Dr. Kevin Williams. Happy Friday.
Hey, Fridays are always good, even with you guys.
Yep, TGIF.
So I'm going to steal an icebreaker that Kirk used for the second paper we discussed today and ask you, what is the favorite place that you lived and a food or custom that you took with you? Kevin, we'll start with you.
Yeah, for me, I think it might be my grad school time, living in New Orleans. You really can't go wrong with the food from there. Anything from gumbo, jambalaya, po' boy sandwiches. But I think anybody that knows me, I have a real sweet tooth. And so bread pudding or bananas Foster are really some of my favorites. I did pick up doing some seafood boils down there. So a lot of crawfish boils. I've tried them a few times here in Dallas. I don't think I'm the best at it, but I think it turns out all right. Nobody complains of being sick or anything along those lines. But what's fun, I guess, with that is that same pot could be used for the seafood boils, but then in the fall, you could use it to deep fry your turkeys. And that comes out pretty good. Oh, and I just realized, you know where ADA 2026 is going to be?
Oh, New Orleans.
It's going to be in New Orleans. I'm ready. And now you have me thinking about food.
Kirk, has Kevin taken you on the tour of New Orleans, the little, sort of ghost tour?
I have not gotten the ghost tour. So,
all right,
Kevin,
well, there you go.
2026 June, I imagine.
Yeah, it's ghost, it's vampires, it's voodoo, it's
Perfect.
Everything, all the above.
All right, Kirk, what about you?
Wow, that's a tough one. Grad school was a lot of fun. I had a chance to do a lot of extracurriculars in a place that I already knew, Indianapolis. But I think the food really comes down here because I learned how to pull pork or to barbecue pulled pork. And that's something that I never even attempted until we moved down here. And now we do it not quite weekly, but a lot.
Yeah.
It's easy to feed a lot of people that way. So
yeah.
So yeah, that's the food side.
Cool. Well, y'all know I'm from New Mexico, so that's kind of a special place for a lot of reasons, and the food is delicious. But I'm actually going to pick Nashville, and instead of food, I'm going to say bluegrass. That's kind of where I discovered bluegrass music. And I wouldn't say that I'm like a total diehard and that I have all of my favorite bands picked or whatever, but we do have a couple musicians that we found when we were in Nashville that we still follow. Actually saw one a couple weeks ago here in Denver. Definitely did not expect to enjoy Nashville, and I really love that city.
I think it shows you can live just about anywhere.
Yeah, for sure.
Yeah.
For
Now,
sure.
Badar, I am a little confused. I think we've had the conversation about Hatch Chili's, and is it the Christmas burrito? Is that right? The red and the green?
Yeah. So, when you go to a New Mexican restaurant, they will ask you, do you want red or green chili? And you can get both if you say Christmas style. So
all right, so I did remember that.
Yeah,
okay.
Exactly. All right, well, I guess we should talk some science. Kirk, why don't you tell us about our paper interviews today?
Yeah, we have some good papers this month. The first article was Diabetes Genetic Clusters and Clinical Outcomes in American Indians. We talked to first author Kaylia Reynolds and co-corresponding author Professor Nora Franceschini. We then discuss the article Sustained Weight Loss with Combined LEAP-2 and Semaglutide Treatment in Mice. We talked to first author Stephanie Holm and corresponding author Christoffer Clemmensen. These are diverse papers, but both were really good discussions.
Yeah, that was a fun one. And Kevin, I didn't get to attend this one, but we interviewed I think, kind of an icon for our Sweet Talk segment.
Absolutely. Yeah. And you both know that November is Diabetes Awareness Month. And so I think it's a perfect time for us to reflect on the progress and the understanding and treatment of this disease. The study of incretins has been a revolution in how we approach diabetes and obesity research and treatment. And today we were able to talk with Dr. Jens Juul Holst from the University of Copenhagen, whose work on GLP-1 and GIP really has laid the foundation for the therapies that are being used by millions of people around the world. And in 2021, Dr. Holst also received the Banting Award, which is the American Diabetes Association's highest scientific honor for his pioneering contribution. So stick around and we'll explore the past, present, and future of incretin research with Dr. Holst.
Yeah, that's always so much fun to talk to him.
Yeah, it was really a good conversation.
All right, well, let's dive in.
Today, we will be discussing the article, Diabetes, Genetic Clusters and Clinical Outcomes in American Indians, which is published in the November print issue. We are joined by first author, Kaylia Reynolds, who is a biostatistician in the lab of a co-corresponding author, Professor Nora Franceschini. Both Ms. Reynolds and Dr. Franceschini are in the Department of Epidemiology at the Gillings School of Global Public Health at the University of North Carolina. Ms. Reynolds and Dr. Franceschini, congratulations on your paper and thank you for being a part of this month's podcast.
Thank you.
Now, before we get started, we like to start with an icebreaker, and we haven't talked about this question yet. So let's start with Kaylia. Can you give me a fun fact about yourself that has nothing to do with science?
I would say a fun fact about myself would be that I do quilting.
Oh, cool.
Okay, very good. And Nora?
Well, I love hiking, and that's probably not
nice.
so exciting.
No,
That's
that's
great.
a good one. There's some nice trails in North Carolina, right?
Yes, a lot of that.
Yes.
Yeah. Nice. All right. Nora, before we discuss your article, we'd like to give our authors the opportunity to highlight any other contributing authors or collaborators on this work.
All the co-authors are fantastic collaborators, and I just want to probably bring up Shelley Colt, that is a geneticist at the Texas Biomedical Research Institute, and she generated the data. And Andrew Morris, that is a biostatistician, University of Manchester, and he also had a great contribution helping, develop the statistical analysis.
And then the other thing that we like to start with is for you to give us an overview of your research program and then maybe touch a little bit how that work led to this specific project.
So my laboratory is actually very broad in terms of, the research. So I'm quite interested in cardiometabolic traits and kidney. So I'm a nephrologist. So that comes with, cardiometabolic is essentially the complications of kidney disease or can be, cardiometabolic can be the cause of kidney disease. So essentially, the whole lab goes around these topics, and I apply genetic genomics tools, essentially identify causes or risk factors in this interaction between kidney and cardiometabolic disease.
All right, Nora, this data was taken from a study that was a longitudinal cohort study. Can you tell us a little bit about the cohort demographics and maybe also about the exclusion criteria used to set that cohort?
So the Strong Heart study is really one of I think the largest study in the US of American Indians. So there's also a study, and IDK has also a cohort of Pima Indians. The Strong Heart specifically recruited American Indians from 3 different areas in the US, and they were trying to sample different tribes. And the goal was to identify cardiovascular risk factors and also establish the prevalence and incidence of the epidemiology of cardiovascular disease in American Indians. So when they started this study, the people still believed the American Indians didn't have cardiovascular disease. So this was a kind of a landmark study in that sense. And there is several phases. There is the cohort, that's the one that we use it in individuals 45 or older. After 3 or 4 visits, there was a family study that was embedded into the cohort. So this is the Strong Heart Family Study. So this study is still being followed. There is multiple visits, it's still very active study, and there is a very large group of investigators interested in studying the health of American Indians.
Great. And I guess, as a sort of a follow-up here, as a non-geneticist and for our non-geneticist researchers, maybe you can help us with some of the critical methodology necessary for this study.
So the most important calculation methodology to understand for our study is the partition polygenic risk score. And a polygenic risk score, or a PS or PRS, is a number that represents an individual's genetic predisposition to a disease. So here we're studying type 2 diabetes. Generally, the higher an individual's PRS is, the more their relative risk is for developing a disease. Here we're calculating partitioned polygenic risk scores, which is a little bit different. This means that instead of representing the endpoint of a disease, we're modeling the risk of a disease mechanism, so the way that you would develop the disease. And so a couple examples here, we have obesity and metabolic syndrome and lipodystrophy. These are all ways that can lead to developing type 2 diabetes. We did not calculate these partition polygenic risk scores ourselves. We got this from a paper by Suzuki et al. Published in 2024, and we applied their weights to our Strong Heart study.
Got it.
So I know this study mostly focuses on type 2 diabetes, but you did find something unexpected with the type 1 diabetes classification. Can you tell us a little bit about those findings and the implications?
So this is the interesting thing. So in the Strong Heart study, there is individuals that report having diabetes, very early age, teenager years essentially. And so there's always this question about they have type 1 diabetes versus type 2. And we do not have, antibodies to do the diagnosis, so the clinical diagnosis. Because this is a cohort study, so if they have that information with their clinician, it's not available within this study. So I think that was an important question if you're going to apply to look at type 2 diabetes mechanisms. I think the first step is actually rule out you have type 1 diabetes, because if we don't find associations, it could be that we have a mix of individuals with type 1 and type 2. So the first step was using, and then cohort is being developed by Sharp and all. And essentially they use controls, normal controls. They use type 1 diabetes individuals and individuals type 2, and they're able to estimate based on the genetic profile, separate these groups very clearly, which is quite interesting. So when we use their score in our data, it was obvious that most individuals are type 2 diabetes. They were not type 1 diabetes based on the this score.
Interesting.
So that's a great segue. So we're going to focus on type 2. You've defined 8 diabetic clusters and examined their association with cardiometabolic traits. What were some of those key associations that you found?
So I'm just going to highlight 2 of the clusters just to represent maybe like a contrasting result.
Yeah, sure.
So the first one would be the obesity cluster. We found increased associations with body mass index, hip circumference, waist circumference, just general traits that are associated with obesity, and contrasting that with the lipodystrophy cluster, in which we found decreased associations with the same traits, which also fits the lipodystrophy profile. So this just goes to show that using these traits, we're able to distinguish between different disease mechanisms that lead to diabetes.
And I guess as another follow-up, was there anything different or similar, I guess, about this cohort that you learned as compared to maybe a population that is derived from Northern European descent?
So, the interesting thing that the clusters actually perform very well in American Indians, which is quite remarkable. It makes sense because most of what these clusters are composed is common variants, and common variants tend to be common across populations, right?
Yeah.
With few exceptions. But I think the main issue that we're not able to identify any associations of these clusters with outcomes, which was somewhat disappointing, but it may make us think about specifically this, maybe some specific component of these clusters that are not specifically useful in this population. I think that was actually our main conclusion was specifically that the Americans were not included in the building and development of these clusters. But just to correct this, is actually the clusters were built in multi-populations. So they have a very large number of East Asians, they have a few Hispanics and African Americans in that cluster, and some Hispanics too, but very small numbers. So essentially, there is a potential, I think, of improvement of this particular methods by using expanding to larger populations. So it doesn't work in American Indians, it may not work in other populations that's not represented when they develop these clusters.
So that's interesting, right? So I'm from New Mexico, and it felt like growing up, I heard the risk was always higher for diabetes. So did this result surprise you? Was there anything that you were able to pull out that was specific to this native population?
So, the type 1 diabetes, I think, as we look at the literature, there is actually much less in American in the population. So that was something that I learned in this process of doing this work. So it's kind of actually a concordant what's being published in the literature in terms of prevalence. And then, as you refer, Hispanics and American Indians, these are populations that are being less studied. There's essentially less samples to work with, and I don't think people are avoiding these populations. These populations, just less data is being generated, and that kind of trickle down to less studies and less knowledge, right?
Yeah.
Kaylia, what are some of the strengths of your approach? And are there any caveats to these studies that you'd like to discuss?
One of the big strengths for our study is definitely using the strong heart study in general. It's a very well-designed study with comprehensive clinical and biomarker data, as well as continual monitoring for morbidity and mortality throughout the cohort. Some caveats would definitely be the size of our study. As Nora mentioned, there's just not a lot of data out there, relatively speaking, to European populations on the American Indian populations. And so there's just opportunity to expand into larger cohorts for that as well.
Yeah, we'll put.
So as we finish off, are there any other key points or thoughts you'd like to leave us with? Or maybe talk about where you'd like to take this work next?
So there's been an explosion of methods out there now and interest in using omics in general. So we're using genotypes. So we're essentially building this based on the genetic susceptibility. But there is other pieces of this puzzle that are not being tapped. And I think the environmental factors or the non-genetic factors are really important. And I think early studies on this clustering use clinical phenotypes. So integration of this clinical phenotypes is one thing that will be very interesting. The other thing is including, proteomics, metabolomics, things like that reflect more your environmental, dietary, and other exposures that you have. So I think the field is developing so quickly, and it's quite exciting. And again, I think there will be not one single answer for this. There will be multiple ways to look at the same problem to try to figure out better ways to predict complications. I think I'm quite interested in that part. Specific kidney disease. I think kidney disease is being quite difficult to find the genetic susceptibility genes or variants. And I think it's time to be creative and look all the tools we have to try to figure out better mechanisms. I think it's quite important research. So we are building on this things being developed by others, and then we always look in the literature and seeing smart ways to tackle, these issues.
Excellent. Well, Dr. Franceschini and Ms. Reynolds, we want to thank you for joining us today and sharing your expertise and discussing your recent work on diabetes, genetic clusters, and clinical outcomes in American Indians. I think Kirk and I want to express our personal opinion that studying distinct populations with increased risk for disease is very important work, especially if we're ever going to get to some sort of point of personalized medicine. So I want to thank you for your efforts on that regard. I encourage the listeners to read the full article in the November issue of Diabetes. Thank you.
Thank
Thanks.
you.
Today we have a full house as I have both my co-hosts with me to discuss November's paper of the month, Sustained Weight Loss with Combined LEAP-2 and Semaglutide Treatment in Mice. We are joined by first author Stephanie Holm and corresponding author Christoffer Clemmensen. Dr. Holm is a postdoctoral fellow in the Clemmensen Group, and Dr. Clemmensen is an associate professor and group leader at the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen. Drs. Holm and Clemmensen, congratulations on your paper, and thanks for being on this month's podcast.
Thank
Thank
you.
you. Thanks for inviting us on.
All right, guys. So before we get started, we like to do an icebreaker question. And since many of us seem to travel from city to city throughout our careers, I thought I'd ask, what's the favorite place you've lived? And what's a custom or a food that you've taken with you when you left? So we're going to start with Stephanie.
My favorite place is definitely Melbourne.
Okay.
Yeah, that was amazing. And what was your second question?
What's a custom or a food that you took with you?
Vegemite.
Oh, that must be the Tam Tam biscuit things. Yeah.
Okay.
But we don't have them in Denmark.
Wow.
Christoffer, I know you're going to say Cincinnati and Skyline Chili, right?
Oh, don't disappoint me here.
You actually ruined that, Dolly. I actually had now mentally prepared exactly that
Sorry.
answer. So you read my mind. I would have said Cincinnati and Skyline Chili as kind of, I guess, somewhat of a joke. But yeah, I don't know where to go from next. Maybe I've lived in Munich, Germany, and the chicken schnitzel is quite good. So maybe that's one way to go about it.
I thought you were going to say your tuna salad, but
That's true. I also have a soft spot for good tuna salad.
Very good.
All right, Stoffer, before we discuss the article, we like to give our authors the opportunity to highlight any contributing authors or collaborators to this work.
Yeah, so I'm happy to acknowledge the collaborators that are also listed as co-authors. So this was really a joint effort between many individuals in my research group, as well as research scientists at the pharmaceutical company Novo Nordisk. So I think this is a really nice example of how combining academic basic research with industrial peptide chemistry can accelerate some kind of translational insight into pathways that we're interested in. And this was by no means like a sponsored study by the company, but this was a shared interest in the biology, physiology, and pharmacology of the LEAP-2 ghrelin pathway. And this kind of then the fruition was really this nice paper that we're really happy about.
Yeah, it's really cool. Why don't you tell us a little bit more about kind of the basic overview of your research program?
Yeah, so our lab at the University of Copenhagen largely focuses on how the brain regulates energy balance. And we're particularly interested in how the brain or the organism as whole defends body weight and body fat mass in context of both a negative energy balance, but also a positive energy balance. And obviously, today, kind of those biological defense mechanisms are really what we're fighting against, both in terms of lifestyle interventions for weight loss, but also the new class of pharmacotherapies that we're all very interested in. And the LEAP-2 project fits quite well into this research program, given that it's targeting one of kind of the most canonical or well-known central nervous system pathways involved in appetite control, the ghrelin receptor pathway. So in that way, this is kind of at the core of our research focus, trying to leverage our kind of neurobiological insights into pharmacological treatment options.
Awesome.
Stephanie, your study is exploring this novel approach to obesity treatment. You combined a lipidated long-acting LEAP-2 analog called LA-LEAP-2 with an established GLP-1 receptor agonist semaglutide. I was curious for our listeners to start off here, and the ones that are less familiar with the ghrelin system and LEAP2, maybe you can describe more about what this system is, maybe touch on its molecular target and why the ghrelin and the receptor pathway has become a newer focus in the treatment of obesity and cardiometabolic disease.
Yeah, ghrelin is, to our knowledge, the only known orexigenic hormone, meaning that it stimulates food intake and weight gain through the ghrelin receptor. And therefore, the ghrelin receptor system has been a pharmacological target for decades for the treatment of metabolic disorders. But no ghrelin receptor targeted therapies have yet translated into promising clinical results for the treatment of these disorders. So it was very interesting that in 2018, the Kaplan Lab and collaborators discovered this endogenous antagonist of the ghrelin receptor, which is LEAD2. So LEAD2 basically blocks the actions of ghrelin, but it also reduces the activity of the receptor. So the ghrelin receptor is constitutively active, meaning that it's active without ghrelin present. So, and I think at this point, it must have been like rather mind-blowing for researchers who have been involved in the ghrelin and ghrelin receptor research for years, that we suddenly have this endogenous hormone targeting system. I've worked with LEAD2 for quite many years now, and we know that it is regulated in an opposite manner than ghrelin. So it's downregulated during the fasted or energy-deprived states and returns towards basal levels following refeeding, especially in mice. And we also know that it is positively associated with BMI, so it's increased in humans living with obesity and also decreases with weight loss, which is also opposite to ghrelin. And of course, there's been a lot of interest into this whole system with LEAD2, and it has also shown some promising effects in both reducing body weight with LEAD2 analogs in rodents and also food intake in both rodents and humans, which I find really interesting. So I believe that these findings have kind of renewed the interest into the research on the ghrelin receptor, as this definitely allows us scientists to leverage the body's own hormonal mechanisms to develop safer and more physiologically relevant treatments for cardiometabolic disorders.
Can you tell us a little bit more about the actual compound that you use, the long-acting LEAP2 compound?
So this compound, as Christopher also mentioned, was a collaboration with Novo Nordisk. So they produced this long-acting LEAD2 compound, and it is actually the same compound that was used in the molecular metabolism study from the Jeff Sigman lab, led by Kripa Shankar. I'm not sure if I pronounced that correctly, but it was back in 2024. And with this compound, we know that it has a similar potency on the ghrelin receptor as native LEAD2, and that it also reduces the constitutive activity of the ghrelin receptor. So that's very interesting. We also know that this compound blocks acute ghrelin-induced food intake in mice, and we have also in our study demonstrated that at least partially it mediates the effects through the ghrelin receptor system by use of ghrelin receptor deficient mice.
So correct me if I'm wrong, but these pathways, GLP-1 receptor signaling and then ghrelin receptor signaling, right? They're distinct.
Yeah.
And how did you arrive at the hypothesis that they might work together?
Exactly at that point, I think. So given the distinct mechanism of action and engagement of different receptor systems and neuronal circuits of GLP-1 based therapies such as Semaglutide and also LEAD2, we asked the question whether there would be any additive effects of the combination of LEAD2 or long-acting LEAD2 and GLP-1 receptor agonist Semaglutide on metabolic outcomes. So that was our second hypothesis of this study.
And Stephanie, I love the description of LEAP2 as the inverse agonist of the ghrelin receptor. You know, so commonly we think about agonizing or antagonizing a system, but as the ghrelin receptor having that baseline activity, this inverse agonist has the ability to really kind of shut off an engine of food intake. And so it makes it a very interesting, novel kind of target, I guess, for potential therapeutic intervention. And, you know, in your study, you tested the effects of LEAP2 signaling on energy balance, both alone and, as was alluded to, in combination with a common GLP-1 receptor agonist, semaglutide. And you found some pretty notable effects on energy balance. So I was wondering if you could walk us through some of those key findings.
Yeah, so first we characterized the long-acting LEAD2 just as a monotherapy for weight loss and food intake. And actually, in contrast to the study led by Krivashenka in 2024, we found that a long-acting LEAD2 treatment actually reduced food intake compared to a vehicle control group in diet-induced obese mice. We also found by use of an indirect calorimetry system that mice treated with long-acting LEAD2 for 14 days preserved energy expenditure despite having a rather substantial weight loss during this period. So in this study, we compared our LEAD2 monotherapy with a weight matched control group, which was matched to the body weight trajectory of LEAD2 treatment. And we saw that LEAD2 treatment preserved or treated mice preserved energy expenditure compared to the weight match group that decreased the energy expenditure. Does that make sense?
Yeah, exactly. And I mean, the effects on body weight that you saw were pretty profound, to where you were getting about a 10% reduction in body fat there, but the energy expenditure seemed to be preserved, as you're alluding to. Why is that important? Why is that a significant finding in your mind? And also, does that reflect maybe some kind of an increased sympathetic tone downstream of LEAP2 antagonism?
Yeah, so first of all, I think it's very important because usually during a weight loss, we would expect to see a decrease in energy expenditure, which was also detected in our weight match control group. So they lost about the same weight, but they also decreased the energy expenditure to defend their body weight, so they don't lose any more weight. And for the mechanisms behind this, we actually don't know whether it's due to like increased sympathetic tone or what it is, but we would really like to go more into details with that.
Also, in your study, you had a treatment design in which you were utilizing either semaglutide or caloric restriction, and then transitioning off to LEAP2 or maybe LEAP2 plus sema, really focusing on a pre-weight loss strategy with a caloric restriction, or semaglutide, and then really strength of your study. And I was wondering if you could comment on that's important for our audience and what it suggests for potential future treatment strategies.
It's sometimes very difficult to explain without like showing the graphs. So I highly recommend to read the paper. But in one of the studies, we pre-treated with semaglutide before subdividing the groups into different other treatments. And in this study, we saw that when, first of all, combining LEAD2 and semaglutide induced a greater weight loss compared to semaglutide monotherapy in that study, which is the study 4. Then we subdivided the treatment groups, and also that was really that we could, at this point when mice were treated with semaglutide, they were in a negative energy balance, and we thought that if we could give LEAD2 at that time after or combination therapy here, we could probably inhibit the drive with weight regain. And in the combination therapy following semaglutide treatment, we saw rather not statistically significant further weight loss, but we didn't see rebound as we did with LEAD2 monotherapy after. Then we also did another study where we caloric restricted the mice before pharmacotherapy. And here we had the same hypothesis whether we could inhibit the drive of weight rebound following caloric restriction, because they were restricted to 30% weight loss, which is a lot in mice. And with the combination therapy and semaglutide, we induced a further research with pharmacotherapy, which was quite interesting to see that we could actually get about 41% or so weight loss with combination therapy of long-acting LEAD2 and semaglutide, and almost the same with semaglutide monotherapy. But what I found really interesting here was that when we stopped the treatment of pharmacotherapy and just let mice stabilize in their body weight again and followed them for the rebound period, we saw sustained effects on body weight in the combination or previously combination therapy treated mice with LEAD2 and semaglutide compared with semaglutide monotherapy.
All right, great. Stoffer, so why don't I think you found something a little maybe, or at least not what you would expect in terms of looking at what semaglutide does to lean mass with the long-acting LEAD2. Can you talk about those findings? And were you able to observe any changes in maybe skeletal muscle endpoints or endocrine regulation?
Yeah, so I mean, we typically measure body composition in all these experiments, and it's always a little bit of a kind of an open question when you want to do this, because it also adds some stress to the animals when you follow weight trajectories, right? So we try and do like a pre-post type of analysis, and sometimes after weight regain, and these snapshots gives us an indication of whether the changes in body weight can be attributed to lean versus fat mass. And some of the studies that we've done imply that maybe LEAP-2 doesn't shred as much lean mass as would be expected from kind of similar weight reducing effects of other type of molecules, such as a GLP-1 or caloric restriction. So it may be that targeting the ghrelin receptor pathway is beneficial in terms of preserving lean mass versus shredding fat mass. But I think we need more studies to be able to say this with more certainty. And we haven't done too much in looking at other endocrine markers to explain this, or even looked at skeletal muscle markers of hypertrophy, atrophy, but it's something we definitely would like to do also going forward.
So I think we're about to wrap up this discussion. Dr. Clemmensen, as we do wrap up, are there any final thoughts you'd like to leave our listeners with, or perhaps is a sense of where you'd like to take this work next?
Yeah, so I think it's been super exciting the last 5 years or so, right? That we've seen this battle both on the preclinical and clinical side on maximum weight loss efficacy. So who can push the most weight loss in humans? We've approaching 25% with the triple agonists. And I think now we're moving towards kind of other virtues of weight loss, including like quote unquote, what is a healthy weight loss? And that not only means efficacy, but also tolerability, right? So how do we achieve weight loss without getting all the adverse effects, or at least dampening those, but also durability? Because we've seen from the semaglutide and tirzepatide trials, once you stop treatment, we see a very similar weight rebound, right? So if we can offset that weight rebound with any type of additional treatment substitution or add-on therapy, I think that could be very valuable, because you also see with the weight regain, you also see all the comorbidities emerge very, very rapidly. And that's, of course, a place to focus on moving forward. So I think that's an exciting avenue of the LEAP-2 work that we're doing also. Maybe we don't see those massive, major preclinical add-on benefits with LEAP-2 and GLP-1, but maybe we actually need to look at the nuances instead, because also we don't know how well they translate. Maybe we actually see these benefits translate quite well in humans. So I think that's some type of note to end on. And then also finally, the more insight we gain onto the neurobiology of feeding and weight regain and the role of AGRP and its plastic changes during a negative energy balance, I think that opens up also new treatment opportunities to specifically target AGRP neurons, because we know with the incretin hormones that we only indirectly target that neuronal subpopulation. So I think that's also a very exciting part of the LEAP-2 story that we will take with us as we move forward with this research.
Well, Christoffer and Stephanie, I want to thank you both for joining us today. You've shared your expertise, and you've discussed this really cool paper, right, on the crosstalk between GLP-1 receptor signaling and ghrelin signaling axis on energy balance. And I want to encourage the listeners to read the full article, which will be in the November issue of Diabetes.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and I'm joined by my co-host today, Dr. Kirk Habegger. Today, we are focusing on incretin hormones. These are discoveries that have completely reshaped how we treat type 2 diabetes and obesity. We're honored to be joined by one of the pioneers in the field, Dr. Jens Juul Holst from the University of Copenhagen. His work has revealed the biology of GLP-1 and GIP and really laid foundational work for therapies now used by millions of people around the world. In 2021, Dr. Holst received the American Diabetes Association's highest scientific honor, the Banting Medal for Scientific Achievement. Welcome, Dr. Holst, and thank you for being a part of the podcast today.
Thank you very much, and thank you for asking me to join you.
As we start this off, we typically start with a little bit of an icebreaker question, and I think we have a larger, big icebreaker question. If you could solve one unanswered question in diabetes or obesity research today, what would that be?
I thought about this, and thinking about diabetes, there's one question that I think is above everything else. That is, why doesn't the beta cell respond to glucose? I'm pretty sure, if we really could understand why it doesn't do this, which is the most important job of a beta cell, to respond with insulin secretion in response to glucose. Why doesn't it do it? And I haven't seen anybody who's been able to come up with a good explanation for this. And I'm pretty sure that, our inability to understand why GIP, the other hormone, doesn't work very well in diabetes, that is because it hits this problem that the beta cell is unresponsive to glucose. GLP-1 also has that problem and eventually will fail if the beta cells really seriously fail. So it won't be effective as a diabetes medication unless there is a little bit of beta cell function left. But apparently GLP-1 has this ability to recruit a G-cube mechanism that GIP does not have. At least that's what somebody think. And that's what rescues it a little bit, but not completely. So the key question remains, why doesn't the beta cell respond to glucose? And I think that's really an important one.
Yeah, that's a great start to our conversation, Jens. You've been part of this incretin story almost from the beginning, right? When you look back at those early days, do you recall a turning point that convinced you that these gut hormones were not just a curiosity, but were actually fundamental to glucose homeostasis and maybe a potential new way to treat diabetes?
So I've been interested in the gut hormones for many, many years now. And I entered this field with a firm belief that the gut hormones were important. In the beginning, we were thinking more of, regulation of the GI tract. And in particular, the focus was on acid secretion, because the focus in those days was peptic ulcer disease. So we were actually looking for something that would inhibit acid secretion. But we're also extremely fascinated by this new concept of the incretin mechanism, which was unknown in those days. So it was a challenge to find out, how was that possible? What was the incretin effect? Were there any hormones that were responsible, and could we find those hormones? So that was the initial impetus for us. So we were firm believers that they were interesting, but from a physiological point of view, and therapy was mainly in peptic ulcer disease, maybe. But it was always interesting to find something that would stimulate insulin secretion. But frankly, people didn't believe that you could treat diabetes with any of these hormones. Because again, the beta cells were sick. They couldn't do what they were supposed to do. And nothing that people had been working on would help them, would do anything good to the beta cells. So why should a gut hormone be able to do this? And sure enough, when GIP was then isolated and found to be a good incretin, very quickly in our laboratory, actually, we were just demonstrating that GIP didn't work in people with diabetes. That was in 1987. So that, of course, supported this idea that you could do nothing in diabetes with a gut hormone. That was completely impossible. So we were focusing on all the other things. And here, appetite regulation was an important feature. It was known since 1973 that the other gut hormone, cholecystokinin, could inhibit food intake in rats. And that caused, a very, very big stir in the field, that perhaps there was some regulation of appetite and food intake from the gut. And that was actually a major focus from the beginning, actually. And that's why we also rather quickly turned into looking at food intake with GLP-1.
You touched on something that I want to circle back to, and that is the reluctance of the scientific community to really hang on to something new, right? You mentioned you felt some resistance. What were your strategies as you're exploring these incretin hormones and their new roles? And how'd you push through that?
I guess we had to convince ourselves first. As you know, we were able to isolate a new unknown peptide from the gut that would stimulate insulin secretion. We were able to demonstrate that. This was back in 1986. And that was, of course, interesting, but it was not a sensation, because there were many peptides that could stimulate insulin secretion. Up there in Stockholm, Jorpes and Muth, they were isolating new peptides all the time, and many of those would stimulate insulin secretion. So it's really when we found out that it also inhibited glucagon secretion that things began to be a little bit interesting, because this was new and different. And as you know, glucagon secretion is increased in people with diabetes and is responsible for a considerable part of the hypoglycemia. So hitting the glucagon target was really something interesting. But again, it took a long time before we were able to convince ourselves that this was a really good idea. And eventually we did a number of studies alone and together with other people, particularly in Stockholm and Sweden, where we were infusing people with GLP-1, and it worked somehow. The studies with the people in Sweden with suadefintage were very, very complicated. We were using the biostator, the artificial pancreas, as they said in Germany, an extremely complicated machine, with infusions of glucose and fluids and glucose and insulin and all that. The protocol was enormously complicated, and the paper that came out of it actually in 1992 in New England Journal of Medicine was so complicated that nobody has quoted it ever since, although it demonstrated quite nicely a stimulation of insulin secretion in people with diabetes. So the real breakthrough was the collaboration with Michael Nauk, first in Göttingen and then in Bochum, and Bad Lauterberg in Germany, at the diabetes hospitals there, and where we were able to show that intravenous infusions of GLP-1 could completely normalize high fasting glucose levels in people during a 4-hour infusion, and that this happened while insulin secretion was stimulated and glucagon secretion was suppressed. This was published in 1993. And that to us was the real breakthrough. And this was actually what spiked the interest of the pharmaceutical industry to enter this field. So it was a true breakthrough.
Building on that, I wanted to switch and talk a little bit about more of the therapeutic applications of these medications and these incretins. And we've talked about a little bit of the implications as it relates to obesity and diabetes, but the role of these drugs or these compounds in areas outside of obesity and diabetes have started to emerge. And I was curious about your thoughts on the role of these incretin therapies in treating multimodal diseases outside of diabetes and obesity.
So when were the first signs of all this? I mean, first of all, the first many years passed by mapping the effects on the GI tract and the mechanisms there, because we also have all these motility effects and effects on the vagus nerve and on all these functions. So it became more and more clear that we had here an interesting hormone that normally would regulate food intake and adjust it to the digestive capacity of the gut, so that you had a control of it. And we used to call that the ileal brake mechanism. So the physiology of this substance was being worked out in some detail, and we understood it, we somehow felt. Then it's really after the clinical application in the large trials, when clinically useful preparations were made, that we were beginning to see some of these, I must admit, unexpected effects on cardiovascular disease and on blood vessels and neurobiology and all that. And they were unexpected, and I really can tell you that for sure, because this whole story really started with the FDA demand of cardiovascular outcome trials of the new diabetes agents. And the first sensational one to come out was the one with empagliflozin, of course, the EMPA-REG studies, that showed for the first time in the history of diabetes therapy that you could do something about the cardiovascular risk of diabetes. So this was really a sensation. And that was in 2015, presented in Stockholm. The audience, I was there, went absolutely crazy when we heard this. We were jumping up and down and standing on the chairs and shouting hooray, and because it was the first time that it was possible to do something really important for the people with diabetes. But immediately the question then came up, which was not answered: would the GLP-1s do the same? And so nobody knew about this. So it was extremely exciting to see what happened with the LEADER trial, which was the first cardiovascular outcome trial, with liraglutide, which was at the time the best and most important of the GLP-1 receptor agonists. And fortunately, the LEADER trial came out with a result on MACE, which was as good as that of empagliflozin, namely a 13% reduction in in MACE in that study, which was as big and as good as the EMPA-REG study. There were more effects on heart failure with the SGLT2 inhibitors, but that picture has turned with the advent of more studies and better drugs, so that now we know that the GLP-1s as a group, and really today, I mean, the only interesting ones to talk about are semaglutide and tirzepatide. And we know that they're associated with quite considerable improvements also for people with the preserved ejection fraction heart failure. So fortunately, it has turned out, and I must admit rather unexpectedly, that this would also apply to the GLP-1 receptor agonists. It was unexpected, as I say, and the time has then gone here by trying to find out why that is. And here there are two extremely interesting observations that have come out. They actually were beginning to show up with liraglutide already. And one was, let me group them like this. It was risk factors for cardiovascular disease. It was noted quite early on that the risk factors showed some improvement. And today they show a lot of improvement with the good ones. And in particular, we have the inflammatory markers that show a very remarkable decrease. And this is particularly true, of course, for CRP levels that are lowered by at least 50% or even 70% in the clinical studies. So that means that the systemic low-grade inflammation that everybody agrees that you have with obesity and also in diabetes, which is considered pathogenic factor for the development of the complications, is affected by the GLP-1s. And there may even be a substrate for this, a biological substrate. And as you know, one of the sites where the GLP-1 receptors are most abundant is in the intraepithelial lymphocytes in the gut. They're completely stuffed with GLP-1 receptors. So you can imagine that physiologically, the hormone is released and interacts with these lymphocytes and then ignites the immune system. So, I mean, this is not something that has been pulled out of thinking. This is something that you can actually demonstrate. So more and more studies are now working with the immune reactions or the effects of the GLP-1 receptor agonist on the immune system. So that's a very, very important thing. The other very important thing in my mind is the fact that the GLP-1 seem to act on the blood vessels, the small blood vessels, the capillaries. And that's where we have some of the receptors. And it's been repeatedly demonstrated that GLP-1s can influence progression of lesions in the vascular system. For instance, atherosclerotic lesions. In various models, you can improve that. And so this really is an important target also for the GLP-1s, it appears. And by that, you can understand that it can have beneficial actions for many organs, not only the pancreas, of course, with the beta cells and the receptors there, but also the liver and the kidneys and the heart and the central nervous system, because they all share the problems with inflammation, and they always have the same kind of circulatory system. And if you can improve that, and that's what appears, then you can understand that they may have this beneficial action. There's an interesting detail here that we shouldn't forget. That's the weight loss, because it's quite well established that weight loss is very, very good for you, also to prevent lesions, also to prevent cardiovascular problems, and also to prevent all the metabolic complications that you have. I mean, it's pretty, and I'm also convinced that this is true. And we've been working now for 25 years with bariatric surgery, where we see this in full bloom, right? That you have these fantastic effects of the weight loss. But there is one study that I cannot forget, and that's the study with albiglutide. Albiglutide is given up now, but it was promoted by GSK as a treatment for diabetes, of course. Before it was closed down, they just finished their cardiovascular outcome trial, and that showed that there was a very nice beneficial effect on cardiovascular outcomes, a base effect which was similar to the others, but no effect on weight. So that's still on the mind of everybody who's working here, that there must be something in addition to the weight loss. And that's what we've been talking about, right?
Right. Jens, I want to pivot just slightly. You have often emphasized that GLP-1 and GIP are both incretins, but that they are not interchangeable, right? So I was hoping maybe you can give us some insight into how you're thinking about GIP with regard to its therapeutic effects.
Yeah. Just to put my head in the nettles here. Since you know so much, then you may know that GIP is not a substance that I would recommend for diabetes therapy, and not for obesity therapy either, because I honestly cannot see any evidence that they will do their job. And there are so many studies that have documented this. And, at the EASD, we were demonstrating how 6 weeks of infusion of GIP to people with diabetes who were in semaglutide therapy didn't do anything to A1C and didn't do anything to body weight at all, even if they were put on top of the therapy. So, I mean, I simply cannot see it. There is a little bit of a rather weak evidence that the presence of GIP may influence the side effect profile of GLP-1s, because that remains a problem, as you know. 10-15% of people taking GLP-1 receptor agonists will have these problems. And unless you are very careful with them, they will also cause the problems. And it may be that the addition of GIP activity has this effect to dampen the side effect profile of the GLP-1s. We need a lot of good studies. It is not good enough to refer to the shrew that can vomit and will vomit with GLP-1, and you can prevent that with GIP, because people are not shrews. I mean, so there's still a way to go here. But I do think that GIP is interesting as a metabolic hormone. And we have been working a lot with GIP. And you may know that we developed in our laboratory a GIP antagonist that could be used for people. So we've been doing something like 25 clinical trials now with that antagonist to try to map the effects of GIP, both in healthy individuals and in people with diabetes. And we've come to an understanding that, yes, you can do something with GIP antagonism. And that, in my mind, my physiological understanding of this, perhaps the major thing is really the regulation of GIP of intestinal blood flow, because it seems that GIP is an agonist will promote nutrient absorption. And in this way, it contributes to the deposition of nutrients in the body. In other words, it promotes obesity. And you can demonstrate this in various ways. And this was the original observation also. And a key figure here could be the stimulation of the intestinal blood flow. And you can reduce that with the GIP antagonist. So right now, we have this idea that this could be one of the mechanisms for GIP antagonism to do something. It's a little bit of a crude mechanism, but it's okay. I mean, if it's there, then it's there. What we don't understand yet is what is the effect on appetite and food intake. And again, there are many beautiful studies in rodents and in mice, fantastic studies, particularly from Munich lately. But again, we are not mice. It is different. They are different.
Right. I agree. It's a fascinating system.
Yes.
And I want to stay on the development of these compounds from preclinical studies and on into the clinic. And, one thing that always fascinates me about the incretin-based therapies is that they didn't really come from this classical molecular genetics playbook where you knock out a gene in preclinical models, you develop obesity in these preclinical models, then researchers try and do the opposite and see if they can reverse that weight gain and develop future pharmacotherapies. With, in preclinical models, GLP-1 receptor knockout mice really don't show that much of a body weight phenotype. But if you use GLP-1 to pharmacologically activate the system, it's a potent driver of weight loss that could be observed in these preclinical studies all the way up into the clinic. And that's why these compounds are so effective. And so I'm curious of what do you think this teaches us about how to approach development of future obesity drugs, future drugs for metabolic disease? I know we touched a little bit earlier on of this early research in this topic met some resistance, and I can only imagine that part of that resistance was based off of that physiology versus the pharmacology of the system. And so how should researchers be thinking about development of these future therapeutics?
Yeah, that's another good question. So one of the big challenges right now is to provide sufficient supplies of these very good drugs and to make them available at an affordable price, which has turned out to be a major problem. And that's where developments really should happen and could happen, and that particularly in the application of small molecule activators of the GLP-1 receptor, even if orforglipron is not a simple small molecule, it's a very complicated one. It's still a small molecule, and that means that it can be synthesized, produced in large amounts, and the technique is kind of available for medicinal chemists in many settings, so that you can imagine that such small molecules can be available in large amounts and at an affordable price. And that will solve some of the major problems that we have right now. Interestingly, in all these new compounds that are popping up every day, when I open my computer, there are some new interesting ones. There was a quadruple one that I was looking at here today, and a pentabile, a 5-acting multi-agonist that was announced also. But they all have GLP-1 in them. So it appears that there's really nothing without GLP-1 as the kind of basic compound, and that to me signifies that we have been able to hit a very important regulator of appetite and food intake here, and that is an extremely important basis for regulating metabolic diseases. And it comes back to what the problem really is. The problem is that we eat too much and we work too little, and we cannot do much about this work problem, this physical activity. I mean, yeah, we can ask people to do it, but we can't do that pharmaceutically, right? But we can definitely do something about the appetite question. So it came up quite early, and it turned out that this was, of course, interesting. And we were looking at it, and we were doing dose response studies. But those response studies also revealed that there were side effects. So all the years during this development, it has been a struggle and a challenge between the side effect profile and the effects. We knew already back in 2001, when we reviewed the field, that the effect was dose dependent. So the more you could give of a GLP-1 agonist, the more you would inhibit food intake. But we also knew that it would cause pretty severe side effects. And, it's killed several compounds, these side effects. Think of something like Semaglutide, which was a wonderful molecule, but not harnessed in the correct way. This slow development, and people were so afraid of the side effects. For instance, at Novo Nordisk, they were also afraid of the hypoglycemia. And I was spending a lot of time with the people out there at Novo Nordisk trying to convince them that this would not present a major hypoglycemic risk. And I really had to travel out there all the time to convince them that this was not a problem. And they were as afraid of the side effects with the vomiting and all that. And, semaglutide was almost dying on the way in its development because they were injecting it into too high doses too early. And it had severe side effects, of course, but eventually it was found out how to handle this. Now it takes half a year to up-titrate these doses, which is what it should be.
And I think this builds on to our final question before we wrap up, which is, as you were going through these investigations, I'm sure you have a lot of hindsight and thoughts on what trainees or junior investigators should be thinking about. And so I wanted to ask you your advice. What would you give to a young scientist that's entering the field today, whether it's about choosing the right questions or navigating collaborations or keeping perspective in a very rapidly changing area like we are in right now with metabolism research, obesity research, and diabetes research? What kind of advice do you give to trainees and junior investigators that are reaching out?
Yeah, so I guess my advice would be to get to learn as much about the real problem as possible. And, only about 10 years ago, people didn't think that it would ever be possible to treat obesity. I mean, this was considered impossible. And, the first compound that was registered for this was Saxenda, which is liraglutide at 3 milligrams. And it was approved because of these quite impressive scale trials that provided a reasonable weight loss, but it was never a success. And why wasn't it a success? Because the weight loss was not great enough. And it was not big enough to really create a difference with the cardiovascular problems, the complications. So you have to understand the problem first, and the problem is huge. There is a huge problem out there. There is a whole quarter of the adult population with a metabolic syndrome, and they run a tripled risk of having myocardial infarction and stroke and death and all that. And that is the real problem. So we have to realize that this is a huge problem. And why do they have this? That's because they eat too much. I mean, that's because we have an obesity problem. So the focus should be on the real problem, and whether they want to try to do this by psychotherapy or whatever they want to do. This is the real problem. This is the challenge, to find out what has happened. Why is it that everybody gets so obese, even us, also in our own country? I mean, everybody can sit in a leaning chair these days and order everything with a smartphone. You don't have to stand up for a single second to make your life running. If that is not, an extreme situation that we put ourselves in, I don't know what is. So, I mean, this is the focus of the problem. We can solve it by working on appetite. And as you know, another extremely interesting thing, there was the reward system. It turns out that GLP-1s also act on the reward system. We just learned today that we have been a paper that I contributed to with semaglutide in alcoholism has been accepted. So it's really a major thing, and it works. I mean, it's true. It has this effect on the reward system. So we've been able to identify a handle to enter this metabolic disease and turn that handle a little bit to try to improve things. It reminds me a little bit of other things that we can do when we have problems with our body. For instance, you and I, we are both wearing glasses because our eyesight is not good enough, but we found out that we could do something about it. So we buy ourselves some glasses, and that helps.
Yes, that's great. Thank you, Dr. Holst, for joining us today and sharing your insights on the incretin story. It's really a fascinating discussion. It's really been a privilege to hear your perspective on where the field has been and where it's headed. We want to remind our listeners to read the article that Dr. Holst contributed on his Banting Medal lecture. It's published in Diabetes, and it's titled Treatment of Type 2 Diabetes and Obesity on the Basis of the Incretin System. You'll find a link in the episode notes for that article. And as always, don't forget to like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.